Assalamualaikum warahmatullahi wabarakatuh. We are from Group 5. We'll present you about the improvement performance of the natural enemies. First, let me introduce you about our member. First, myself, Ilham Ardimas Briantama, NIM 111, Ogi Prayoga with NIM 121, Sara Vanesya with NIM 131, and the last is Titin Andriyanti with NIM 13 and 5. The next is background. Background or the topic we are discussing today is the natural enemies have been long and widely used to control the disease and pests in crops. These natural enemies include parasitoid, predators, and the pathogen to control the pest, as well as competitor or antagonists to control the pest.
Genetic manipulation. Genetic manipulation of natural enemies of insect pest offers promise of enhancing their efficiency in agricultural cropping system. Genetic improvement project with natural enemies of insect have been conducted for improved climatic tolerance, improved host finding ability, change in host preference, improved synchronization with the host, insected resistant non-diapause induction of Thelytococcus reproduction, monogenetic traits are controlled by single gene, are easily liable to change over polygenic traits. Such manipulation of biological control agents seems to be logical way of controlling pests in field conditions. Next, the role of biotechnology. First, biotechnology need to be applied to fulfill the aim. The first one, cryopreservation. Cryopreservation is the process of cooling and storing cells, tissues, or organs at very low temperatures to maintain their viability. For example, the technology of cooling and storing cell at a temperature below the freezing point 196 C permits high rates of survivability of the embryo of the point melanogaster cell upon thawing. Next.
The second way is through the altering biological properties, which are shortening the development rate, increasing progeny production, change sex ratio, extending temperature and relative humidity tolerance, altering host or habitat preference, and those things are to improve the effectiveness of biocontrol agents. So I will give you the example of extending temperature and relative humidity tolerance. The example of the point is the flea found in California, the United States of America, which is Echeria purchasei. They attack the orange crop in there, and they decided to introduce a beetle, which is Rhodolia cardinalis, and I found that the climate of those two regions is different because California is hotter than Australia. It proves that the beetle Rhodolia cardinalis, through biotechnological process, can be adapted in California, the United States. The next one is novel gene transfer. Gene transfer is the process by which a gene from any source can be introduced into natural enemies or tissues. Gene transfer technology can be used to manipulate cells for scientific research as well as for commercial purposes. So one of the method of the gene transfer is maternal microinjections. Maternal microinjections is they did the DNA transfer from the adult female to the egg. The result for the egg itself is they have a high level of survival and transformation.
Example, this replica plant function as a shelter for natural and beneficial insects, or natural enemies and parasitoids. This method has been tested in Vietnam and Philippines by planting flower and vegetable on rice bed. Planting flower and vegetable is expected to attract bees, wasps, and other beneficial organisms, so that when parasitoids come, there will be natural enemies in the land. Is that with the development of insect will be managed and not increase above the threshold. Conclusion: genetic manipulation of natural enemies of insect pests offer promise of enhancing their efficacy in agriculture cropping system. Genetic transformation can be used to improve modify the genome of natural enemies, change sex ratio of parasitoids, cryopreservation, develop genetic linkage map, identify biotypes, improve artificial diet, monitor establishment and diversal parentage analysis and genetic change.